Hi everyone, I'm Sian Wilson and I'm in the first year of my PhD at King's College London. I'm going to talk to you today about how we've used diffusion-weighted imaging of the developing fetus in utero to characterise white matter development. So why we're interested in the fetal period? It's a very unique time frame in brain development due to the rapid changes at the micro and macro level on a weekly basis. These changes are underpinned by the biological processes of neuronal migration, cellular organisation and myelination. This is a critical juncture in development, as structural foundations are laid for a functioning brain. We also know that the intrauterine environment is highly unique and specialised for facilitating brain development, because in cases of preterm birth, there is a greatly increased risk of developing neurological disorders. In order to understand why white matter is susceptible to damage over the second to third trimester, we need a trajectory to describe its normative development in healthy fetuses in utero. This has been enabled by the Developing Human Connectome project, scanning 1,000 neonates and fetuses with multi-shell high angular resolution diffusion MRI, acquired with the parameters shown in blue. This project has developed methods that overcome the unique challenges of fetal neuroimaging, such as the large amount of motion to correct, low signal-to-noise ratio due to the distance of the fetus from the coil, and the fluid surroundings. These limitations have meant that many previous studies have high dropout rates and have ended up with smaller cohorts. Using a bespoke pipeline of data-driven dynamic distortion correction and slice-to-volume motion correction, we're able to move from the image on the left to the image on the right at these different B shells. With these methods, we've been able to estimate tracts in 113 out of 151 reconstructed volumes. Traditionally, the tensor model was used to estimate white matter pathways, and fractional anisotropy maps, like the ones shown here, were used to represent the amount of diffusion in a particular direction. We looked at these maps for all subjects and found that as the microstructure of the brain matures over increasing gestational age, we saw a linear increase in the fractal anisotropy, consistent with previous findings. We also see linear increases in brain volume with gestational age, verifying that our data are typical of the fetal period. The results I'll reveal shortly show how our tract-specific trends are noticeably different to these changes in brain volume and whole brain FA. However, there are some important caveats to remember with this model. Those are the inability to discern crossing fibre bundles and the partial voluming effects when a single voxel contains multiple tissue types. Both of these can affect FA values. An alternative method that's been developed to estimate fibre orientation density functions is constrained spherical deconvolution. The principal direction of diffusion is represented by an ellipsoid that you can see here on the zoomed in version, and interhemispheric connections can be inferred, as well as we can infer transient radial organization. So the pipeline we use to estimate tracts is as follows. We map out the starting points, so for example with the corticospinal tract, that was the cerebral peduncle, waypoints, the posterior limb of the internal capsule in the left and right hemispheres, and exclusion zones, mid-sagittal slice here. And these constraints guide the probabilistic tractography algorithm to estimate the tracts in fiber orientation density function templates that were constructed from averaged fiber orientation density functions of different subjects within a particular gestational age week. We then use nonlinear registration to transform the tracts from the age-matched template into native subject space and thresholded them to reduce the number of spurious streamlines. We characterize changes in the microstructure either using traditional tensor metrics, fractional anisotropy and mean diffusivity, or a novel, more complex method, multi-shell, multi-tissue constrained spherical deconvolution, to address the partial voluming caveat of using the diffusion tensor method. We focused on 4 different tracts, which are documented in the clinical literature as being susceptible to damage over the perinatal period. These tracts were also selected because histology suggests they have different developmental trajectories across the second and third trimester. For example, the corticospinal tract and corpus callosum have been noted to appear around 13 to 15 weeks, the inferior longitudinal fasciculus at around 17 weeks, which only myelinates close towards the third trimester, and the optic radiation, which only reaches the cortical plate at 22 to 25 weeks. We found with the optic radiation it was difficult to track in the template at 22 weeks, consistent with what histology suggests, but more streamlines were apparent 10 weeks later in the 32-week template, which is also consistent with ex vivo findings. So we calculated tensometrics, fractional anisotropy, and mean diffusivity for each tract, and plotted these values against the gestational age of the subject. We then used the Ikeki information criterion to identify which degree of polynomial best described the trends. We found the majority were fitted by a second-degree polynomial, or in the case of the corticospinal tract mean diffusivity, a simple linear downward trend. The dotted lines above and below represent the 95% confidence interval. Previous literature has found linear trends in FA and MD, with FA increasing towards term in a linear way and MD decreasing towards term. However, these studies have often had a smaller range and often began at 30 weeks, which does accord with our findings as we see linear increases from 30 to 38. These studies have also often only had a maximum of 30 subjects, so it's possible we were able to pick up a more intricate relationship with this larger cohort size. We see a dip in the polynomial around 30 weeks consistently in these tracts. There are several processes that might influence FA values which transition around this time point, such as the regression of radial glia between 26 to around 34 weeks, accompanied by the emergence of white matter connectivity. The recruitment of mature oligodendrocyte progenitors is also believed to occur at around 30 weeks and has been demonstrated to contribute to FA values in previous studies. Both of these processes also occur in regional patterns across the brain. We also found differences in the genuine spleen of the corpus callosum, according with ex vivo studies that show these two regions of the corpus callosum develop and myelinate at different time points. We fitted a second-degree polynomial in the case of the genu, the FA and the MD, whereas the splenium was better described by a linear relationship. To overcome the limitation of the diffusion tensor model, we modeled the diffusion-weighted imaging data using this multi-shell, multi-tissue constrained spherical deconvolution, exploiting the unique p-value dependencies of tissue and fluid to estimate a multi-tissue orientation distribution function. This more complex approach to modeling diffusion-weighted images improves the precision of fiber orientation estimates at tissue interfaces. We used it to assess how changes in fluid density and tissue anisotropy correlated with tensometrics. When we looked at the partial correlation between MD values and the CSF fraction in the voxels, there appeared to be a strong linear relationship in all of the tracts between the mean MD value and the fraction of CSF, verifying that the reported MD is actually a reflection of fluid density in the voxel. We represented the tissue anisotropy by extracting the second intensity band of the power spectrum in the white matter orientation density function, and this positively correlated with fractional anisotropy values sampled along the tracts. We also found consistent relationships in all of the tracts, suggesting that the tensometrics did in fact represent differences in underlying neurobiology. So what we can conclude from these findings is that our second-degree polynomials were different to the linear trends that were noted in previous literature. These trends are influenced, and FA and MD are affected by many different underlying processes. These could be biological, such as neuronal migration, myelination, cortical layering and folding, and axonal organization, all occurring simultaneously over the time frame we are analyzing. But there are also methodological considerations. For example, fetal motion artifact varying over gestational age, signal-to-noise ratio varying over gestational age, and bundle segmentation. As the brain matures over the second to third trimester, the tracts that were passing through the brain may have been going through different tissues. We found unique developmental trajectories specific to each tract, which accords with histology, and we also found, when we used the multi-shell, multi-tissue model, that our tensometrics did seem to reflect properties of the underlying microstructure. So thank you very much to all the mothers who consented to being scans, to the DHCP collaborators from Imperial and Oxford, and to everyone who collaborated to this work. I'll now take questions.
Okay, so hello. I'm going to present my study entitled High Temporal Resolution Longitudinal Observation of Fetal Brain Development, a baboon pilot study. So what we're interested in is cortical folding, and there are several reasons to be interested in cortical folding. One of them can be that it provides biomarkers for a number of pathologies. The problem with cortical folding is that it varies a lot across individuals. And if you look at cortical folding patterns as illustrated here, they are really extremely variable across individuals, to the point that it is difficult to define normality intervals and to detect abnormalities. So if we want to understand that variability, we know that we have to look at cortical development before birth in the second half of pregnancy, where folding and variability emerges. Precisely during that period, cortical folding patterns are becoming variable across individuals, and that's why we must look at development in a longitudinal way and not cross-sectional. Also, because the folding changes at a fast pace and we do not know the timing of all the events, we need to observe developments with a high temporal resolution. Unfortunately, so far there is no database, MR database, that fulfill those criteria. And actually, it is not possible to scan healthy human fetuses longitudinally in such a repetitive way. Therefore, we decided to use non-human primates, and within non-human primates, baboons are a very good choice. First, because they are the largest monkeys that we can scan, and when it comes to scanning the fetal brain with MRI, the size is very important. Also, they are the most gyrified, and gyrification is what we're really interested in. And finally, although they show a reduced variability compared to humans, they still exhibit some variability of folding patterns, as you can see here on those three examples, if you look at the arcuate sulcus in the frontal lobe. For baboons, the gestation time is just above 26 weeks, and as humans, it is during the second half of gestation that folding develops. Fetal MRI has become a very strong topic with humans, but it has been also performed with non-human primates, as shown here, on the right with macaque, and on the left with that study on baboons by Peter Koshinov and colleagues. That was the starting point for our own study. So given the complexity of the acquisitions we have to perform, we decided to first assess the feasibility by performing a pilot study. And we scanned a female baboon, pregnant female baboon, once a week for 10 weeks during the second half of her gestation. The goal was to fine-tune all the details of the acquisition protocol in order to get not only anatomical MR images, but also diffusion and functional resting state with a good enough spatial resolution and with a reasonable time, typically below 1 hour. That reasonable time is very important parameters because that's the time we're going to keep the monkey anesthetized. And anesthesia is a key in such a project because we know that repetitive exposure to anesthetic agents can be neurotoxic and disturb normal development. Therefore, we found the only known non-neurotoxic anesthetic agents during pregnancy that still maintains a good level of sedation, and that is dexmedetomidine. In terms of acquisition, we must obviously maintain a good signal-to-noise ratio and a good spatial resolution, while being the least possible sensitive to fetal movements that we cannot control, and that is always an obstacle in fetal imaging. And I would like to stress that the logistics of such a project are very complex, starting with the transportation of the animal that must be done very carefully every week in order to avoid the risk of spontaneous abortion. Also, the large number of people involved must be available every week, once a week for 10 weeks, which is a strong constraint. And finally, we want to assess the effect of anesthesia and check that the development is still normal, and that will be done for 6 months after birth. In terms of acquisition, for anatomical and diffusion images, we chose a typical strategy in fetal imaging, which is to use 2D ultra-fast sequences with a good resolution in plane and thick slices that will be then post-processed to reconstruct 3D isotropic images using super resolutions algorithms. We perform acquisition on Siemens Magnetom Prisma 3 Tesla scanner. And here are the detail of the MR sequences that we finally kept. And I would like to stress that for anatomical MRI and diffusion MRI, we perform acquisition along 3 different orthogonal orientations, twice per orientation. And that redundancy of acquisition is what allows the super resolution reconstruction afterwards. Our total acquisition time is below 1 hour. And we successfully performed acquisition for all of the 10 weeks, except one, because at week 19, there was a hardware failure that prevented the acquisition. So here you can see the results of anatomical images, and you can see for the 9 acquisition sessions. And you can see that first, the signal-to-noise ratio and resolution are good enough to observe gyrification precisely. And you can see that that gyrification goes from a very smooth cortex with few folds and a simple geometry to a very complex geometry and a very folded brain for the last acquisition. For diffusion images, I'm showing here the reconstruction of fractional anisotropy from the diffusion images. First, I would like to say that this has been done with the scanner software, reconstruction software, so it's not optimal at all. But nevertheless, you can see typical features of the developing brain with here visible at the beginning, at 15 weeks, for instance, a very anisotropic cortex due to the columnar organization, and very few white matter tracts, such here as the cortical spinal tracts or the corpus callosum that is also visible. And towards the end of the gestation, the cortex is much less anisotropic because it has matured, and you can see the emergence of much more white matter tracts visible with fractional anisotropy. And finally, I'm showing here a sequence of resting state functional MRI at 15 weeks GA, where the resolution of the image is actually quite coarse compared to the size of the brain. I mentioned before that we want to reconstruct images with a 3D super resolution algorithm. This is still work in progress, but nevertheless, we started reconstructing images, and for that, here an example at 23 weeks GA, we reconstructed using the algorithm provided by Michael Ebner, available here. We reconstructed a 3D isotropic image at 0.5 millimeter of resolution, which is higher than the initial in-plane resolution of the 2D acquisitions. And here you can see the cortical surface reconstructed from a manual segmentation that has been performed by Katia Eyre and Roberto Toro, who I would like to thank for that. So now what comes next? We assessed that the feasibility of such a project we assessed that feasibility. And the project is to now build a database with 10 subjects and 10 time points per subject. And that database will be released to be shared. And we will share not only the 3D reconstructed anatomical and diffusion images, but also the resting state images, and the segmented anatomies and pre-processed diffusion images to guarantee that many labs can actually exploit those images. Acquisition will start next fall and will be spread over the course of a year and a half, so stay tuned for more results. Finally, I would like to conclude by thanking my co-authors, but also the people from the animal facility and the primatology center who were a great help in setting up such a project. Also, I would like to thank again Katia Eyre and Roberto Toro for segmenting our image with their great software, BrainBox. And I'll conclude by thanking the Amidex Foundation at Aix-Marseille University that funded that project. So thank you for your attention.
Hello, thank you for the opportunity to speak today. I'm going to be presenting data from the Developing Human Connectome project on typical and disrupted functional connectivity across the perinatal period. The Developing Human Connectome project provides a large imaging data set of term and preterm born infants to study how brain connectivity is formed from 20 to 44 weeks of postmenstrual age, 40 weeks, of course, being the time of normal birth. This is a critical period in which the brain is wiring and beginning to work together as a system for the first time, setting the scene for later life development. So far, over 1,000 subjects have been scanned. These images can be used to map changes in brain morphology and tissue properties. For example, here we show rapid cortical expansion and myelination in infants scanned after birth from 32 to 40 weeks of postmenstrual age. Many of the subjects are also scanned before birth, as Sian Wilson showed us in her talk earlier in this session. This data is provided as an open source repository for researchers around the world to use as an atlas of normal and abnormal variation in the developing brain to address key questions such as how and when does connectivity assemble in the brain? The example shown here is structural connectivity analyzed using graph theory. What are the genetic and environmental influences on this process? Can early differences in connectivity help explain the mechanisms behind currently intractable neurodevelopmental disorders, such as autism? In this talk, I'm going to present some of the resting state functional MRI acquired in infants around the time of normal birth. As you know, the brain's spontaneous activity at rest can be spatiotemporally decomposed into resting state networks, which are reproducible across populations. These can be identified in preterm infants from as early as 26 weeks of postmenstrual age. Taking the example of the default mode network, this schematic depicts its putative early development, but until now we've not had large enough data sets to characterize the maturation of these networks around the time of birth in detail. So we wanted to address the following research questions. Firstly, how mature is the functional organization of the brain around the time of normal birth? Is there an adult-like network architecture already established, or is it still under construction? Secondly, what is the effect of preterm birth on resting state network development? We know that preterm infants are at increased risk for developmental disorders such as intellectual disability and autism, and even adult onset psychiatric disorders. Might these have their origins in abnormal brain connectivity in early life? 337 babies were included in the study, 65 born preterm. Whether they were born term or preterm, all were scanned at term equivalent age, and just one imaging time point was used for each infant. Scans took place in a customized environment for neonatal imaging. Infants were fed and comfortably positioned in a vacuum jacket prior to scanning to promote natural sleep. 2,300 volumes of high-resolution bold fMRI were acquired for each infant over 15 minutes. Full details of the pre-processing pipeline are available in this preprint paper. Just to highlight two important elements from this. Firstly, retrospective motion correction was built into the pipeline, which minimized the number of subjects we had to exclude for motion. And secondly, data were aligned to a standard space using T1 and T2 structural images acquired in the same session. So the first step in the analysis was to define a normative set of networks in the term-born infants scanned at 43.5 to 44.5 weeks of postmenstrual age, using independent component analysis by temporal concatenation across subjects with FSL melodic. The output was a set of group-level maps or independent components representing each resting state network. The next step was subject-level analysis in the remaining 313 infants. For this, we used dual regression, which took us from the group-level maps to a map of each network in each individual subject. This enabled us to then test for the effects of patient characteristics on the networks using general linear models while controlling for sex and motion as covariates, and we corrected for multiple comparisons with permutation testing. So these are the 11 networks we identified. The 5 on the left included primary motor or sensory cortical areas. We categorized these as primary resting state networks. The remaining 6 we categorized as association networks. The easiest way to visualize these is on a cortical surface in a winner-takes-all postulation. So this is the brain of a single subject scanned at 42 weeks of age. And then the group level networks are projected to the surface. So this network encompasses the medial part of the primary motor cortex. This is the lateral part. This encompasses primary somatosensory cortex. This is an association network, which includes the premotor and supplementary motor areas. This is the primary visual cortex and higher order visual processing areas. This network includes primary auditory cortex. This is an association network, which includes both Broca's area and the extended Wernicke's area. And the last 3 are all association networks in areas serving higher order executive control and attention functions in the mature brain. So next, I'd like to present the subject level results, focusing first on network maturation in the term-born infants only. These maps show average network structure week by week according to age at scan. So you can see all the networks are there at 38 weeks, but there's a subjective impression of more conspicuous maturation week by week in the association networks. Next, we evaluated the effect of age at scan as a continuous variable while controlling for sex and motion. So here, the red-yellow maps show regions where functional connectivity significantly increased with age, and the white lines show the outlines of the group-level networks, thresholded to Z value of 3. So we found areas of increasing connectivity with older age at scan across these 4 networks, which were all higher order association networks. We didn't find significant changes with age at scan in any of the primary networks. To further quantify the effect of age at scan, we looked at the relationship between age and a derived parameter we term core network strength, defined as the mean beta parameter value for each subject within the area bounded by the white lines on these figures. 3 networks showed a positive correlation between age at scan and core network strength, 2 of which are shown here. Next, I'd like to focus on this result in the frontoparietal network, which is here in dark blue, comprising cortical regions associated with coordination of eye movements. We know from tracer studies in primates that the subcortical projections of these regions converge on the caudate nucleus in the basal ganglia. So our finding of increasing functional connectivity of the caudate nucleus within this network with increasing age at scan, as shown here, indicates active maturation of the oculomotor corticostriatal system at this period of development, a time when we know the ability to fix and follow on a visual target is emerging in babies. So to summarize and discuss this section of the results, we found differing trajectories of maturation in resting state networks at normal term age. Networks encompassing primary cortical areas appeared in an adult-like configuration from the earliest ages studied, while the association networks appeared fragmented compared to the adult brain and were undergoing active maturation over this period of development. And this mirrors the heterogeneous timing of maturation in brain structure. In this figure, derived from post-mortem histology over a century ago, the black areas are myelinated at the normal time of birth, and you can see the primary networks overlap with these regions, whereas the association networks, which are still maturing, lie in the areas that myelinate later. Next, we looked at the effect of preterm birth, while controlling for postmenstrual age at scan, sex, and motion. These maps show group differences in functional connectivity between term and preterm born infants. Blue indicates areas of reduced functional connectivity in the preterm group. So as you can see, preterm birth was associated with extensive impairments of functional connectivity at term equivalent age across all the networks studied. Again, we quantified this further with our derived measure of core network strength, representing average within network connectivity in the network core. Term-born infants are represented in the bars on the left and preterm-born infants in the bars on the right. Core network strength was 23 to 41% reduced in preterm infants across the 11 networks. Finally, I'd like to focus on this result, which we didn't expect to see. We found the preterm infants showed increased connectivity of the superior parietal lobules within the lateral motor network compared to term-born infants. This is a region which becomes increasingly connected in the lateral motor network at older ages in the term-born infants. So this seems to represent abnormally accelerated development of parietal connectivity within this network in preterm infants. The superior parietal lobule integrates visual and somatosensory inputs to encode limb configuration and position in the immediate environment. If we think about the environment a preterm infant has experienced by term equivalent age, it is very different to the environment experienced in the womb. One of the differences is the somatosensory experience of moving the limbs outside of amniotic fluid around the body and against gravity. Might this result in these connectivity differences, or is it a compensatory mechanism for impaired connectivity elsewhere? Long-term developmental follow-up will help us understand the consequences of these changes which occur at this critical developmental period in preterm children. So in conclusion, we found a primary to higher order maturational sequence of brain development in healthy term infants. Preterm birth was associated with widespread impairments in functional connectivity and abnormally increased connectivity of the superior parietal lobules within the lateral motor network. Please download our preprint paper, which is available on bioRxiv via this link. And please also download the imaging datasets, which are all freely available to researchers at this website. There'll be many exciting opportunities for future work integrating this imaging with genetics and developmental follow-up. And finally, a big thank you to the many research staff who helped acquire and process the data for this project, and most importantly, to all the families who took part. Thank you for listening.
Hello everyone, my name is Huang. I come from Department of Radiology and the Biomedical Research Imaging Center, University of North Carolina at Chapel Hill. It's the first time for me to join such a meeting, and it's my great honor to present my my research on discovering developmental patterns and the regionalization of cortical myelin during the first 2 years. Human brain development is a complex and long-lasting process that begins during the first 2 weeks of pregnancy and lasts until the end of adolescence. Evidence shows that early brain development is dynamic, nonlinear, and region-specific. The picture shows the dynamic development of cortical thickness from birth to 2 years old. In another study, Huang investigated the infantile developmental regionalization of cortical thickness, which describes regions naturally formed during the dynamic development of cortical thickness and differs marked from conventional anatomical populations. Many works have also been done to explore the significance of early brain development. It has been improved that early brain development is critical for literal cognitive outcome and vulnerable to early neurodevelopmental disorders. However, due to the poor imaging contrast and dynamic appearance, lacking of high quality and processing technique for infantile brain MRI, exploring early brain development is still challenging. Brain development is also characterized by myelination. It's one of the most significant index of brain maturation and is often a prime correlative of speech and language. It plays an essential role in learning and returning new practical skills and is closely linked with the evolution of cognitive and behavioral function. In Bozak's work, neonatal myelin map PUVIC was shown on the inflated cortical surface. It can be observed that myelin content in neonatal brain increases with developmental age. In another study, Weiss and his teammates presented cortical myelin maps for 4 primates and the mouse. From the picture, we can know that each primate species has 6 heavy myelinated regions, from which we can see the development of myelin from the perspective of evolutionary. Taken together, checking developmental patterns of myelination during infancy is of great importance in understanding early brain development. Motivated by this, we tried to compute the first spatial temporal dynamic maps of cortical myelination and discover the cortical regionalization based on developmental patterns of myelination. To do research, 433 longitudinal T1 weight and T2 weight MRI scans were acquired from 209 healthy infants during the first 2 years from the UNC Baby Connectome project. The dataset contained 112 females and 97 males. From the picture, we can clearly see the distribution of longitudinal scans. Each point represents a scan at its scan age. Each line represents one subject, with males in blue and females in red. All images were processed using infantile dedicated surface analysis pipeline. After intensity homogeneity correction, circumscribing tissue segmentation, and left-right hemisphere separation, topological correct and geometric accurate inner and outer cortical surface were reconstructed. After surface reconstruction and spherical mapping, all spherical cortical surface were co-registered into a common space based on cortical folding attributes, such as mean curvature and average convexity. After registration, all spherical surface were resampled using the same mesh tessellation to establish vertex-wise cortical correspondence across all subjects.
I think we might have to start over now.
Okay, we have to.
Is that okay?
Can I restart it? I clicked the wrong button.
We can start from that slide that you left off on.
Okay.
So just go ahead and start. I'll count you down from the slide that you just started, that you were on.
Okay.
Sheet.
So I can continue with it. So should I continue with this?
Let me go ahead and All right, you ready?
Yes, I'm ready.
All right, 5, 4, 3, 2.
Myelination was a computer based on the method introduced by Glasser in Journal of Neuroscience 2011. After registering T2 weight image to the T1 weight image, the ratio of T1 weight to T2 weight signal intensity in each voxel was computed. For each voxel in the native mesh middle surface, ribbon voxels looks like this, was selected within a cylinder between the inner and outer surface. The cylinder had a height and a radius equal to the local cortical thickness value and was centered on the vertex. Then the ratio of T1 weight to T2 weight signal intensity in the ribbon voxels were averaged to serve as the myelin of the vertex. At last, myelination of each vertex were mapped onto inflated surface and the middle surface. Now let's say on the middle surface, after obtaining myelination of all scans, the generalized additive mixed model was adopted to fit the developmental trajectory of myelination in whole brain. As the equation shows, GMM uses additive non-parametric functions to model covariant effects while accounting for over-dispersion and correlation by adding random effects to the additive predictor. Based on the development of myelin, we further tried to create a cortical pethylation map. We further organized the cortical myelin map of each scan into a column and sort our column by ages. An affinity matrix was computed by correlating the developmental features of each pair of vertices on the cortical surface. Then all vertices were clustered into K groups using spectrum clustering. Now, let's say the results of our research. Overall, the myelin map appear bilaterally symmetric, so we just present the results on left hemisphere. The red dots on left figure shows the development of average myelin content changes with age, while the blue curve is the developmental trajectory fitted by the generalized additive mixed model. The lateral view and the middle view illustrate the dynamic changes of cortical myelin map from 1 to 12 months old. To better present the results, we put the myelination of all ages on the same chart, so we can see the developmental patterns as a whole. We also evaluated the lightly and heavily myelination regions of each month. From the picture, we can see the overall spatial pattern of heavy and lightly myelination are largely present at 1 month. Regions of heavy myelination include the central sulcus, middle occipital lobe, auditory cortex, and the sylvan fissure. While light myelination is found in the inferior parental, frontal, and lateral temporal cortex, with which are consistent with that in adults. After clustering of voltages into K groups using spectrum clustering, what that means appropriate cluster numbers according to Sihoit coefficients. The figures show the Sihoit coefficients of clustering results with different cluster numbers, where higher values stand for better clustering quality. From the picture, we can see that a stable plateau is found from 11 to 18, while the local maximum is reached at 12. We thus adopt the cluster number 12 as our pastillation map. As shown in figure, the discovered developmental-based pastillation map exhibits bilaterally symmetric patterns and corresponds to structurally or functionally meaningful specialization. The correspondence are shown as follows. Taken all together, we can summarize our contribution of the research into three parts. First, developmental patterns and the regionalization of cortical myelin during the first 2 years were discovered. Second, cortical myelination maps during infancy present dynamic developmental patterns similar with adults. Third, cortical pathways based on developmental patterns of myelin are meaningful. That's all. If you have any question, please contact me through the emails. Thank you.
Hello, everyone. My topic here is the subgrouping structure of newborns with heterogeneous brain-behavior relationships. One of the most important goals of human neuroscience research is the delineation of robust brain-behavior relationships to develop brain-based biomarkers for behavior prediction. This is especially important for infants, since subtle brain changes during infancy may lead major deviations later in life. However, the reported associations are often weak to moderate. This is because instead of homogeneity, many recent observations suggest heterogeneity of brain behavior relationships. For example, 2 parallel studies from our own group conducted by Elton found that beyond categorical differences in brain functional connectivity, children with attention deficit hyperactivity disorder and autism spectrum disorder showed contrasting brain behavior relationships compared with normal controls. Moreover, in John's paper, sex-related differences in brain IQ relationships in a cohort of control adults were also reported. Therefore, previous results suggested that there might be heterogeneity at the brain behavior relationship level, even within an otherwise homogeneous population. So, we have the hypothesis that different subgroups of infants may possess qualitatively different brain behavior relationships that could ultimately contribute to divergent developmental outcomes. In this work, we sought to take the first step to determine whether there exists brain behavior relationship heterogeneity in a relatively homogeneous control infant sample. So we included 81 typical developing units from the UNC early brain development. After preprocessing, AL template in unit was used to generate the connectivity matrix. The group mean connectivity matrix should here meet our expectation. The basic idea of relationship level heterogeneity is that if there are subgroups showing different relationship within one population, we can never detect using one group fitting. But after we separate the subgroups, we can detect strong relationships. Here's how we detect. With the initial 2 vertical lines, the dots will be assigned to one of the lines which has the shorter dotted line distance, yielding 2 groups of dots. These 2 groups can update the 2 lines using linear regression. Confounded variables were included in the regression. Then, these steps will be repeated for a certain number of times. With this iteration process, the 2 lines will be convergent, which define the final subgrouping structure. The mean R-square from the 2 regression lines was used as the subgrouping performance. Then, for each connection, a non-parametric permutation test was used to assign a p-value for the subgrouping performance. The goal of the permutation test here is to say how this subgrouping is better than the randomized organizations. P less than 0.05 was used to define the significance. Here are 3 examples of homogeneous and heterogeneous connections detected. Across the whole brain connections, there are 343 connections detected showing significant homogeneous one-group connectivity IQ relationship. 334 connections were detected to show two-group heterogeneous brain behavior relationships. If we fit the heterogeneous connections in one group, the mean R square is much lower, but if we fit these connections with the detected subgroups, the mean R square is much higher. Based on the connection levels subgroups, the difference values of dotted line distance of each subject's data point to the 2 regression lines were calculated the subject's level subgrouping profile vector and used for the whole brain level clustering. Based on the Davis-Baldwin criteria, the 81 units were clustered into 2 subgroups named as S1 and S2 at the whole brain level. The results demonstrated significantly higher within subgroup similarity than between subgroup similarity in their connection level subgrouping profiles, which support the existence of whole brain level subgroups. When examined against the 6 control variables shown here, the 2 subgroups did not show any significant difference. However, when examined behavior outcomes, S1 S2 showed a significant difference in 4-year IQ. The S1 showed higher IQ than S2. For the subgroups S1 S2, they showed similar functional connectivity pattern. No significant difference was found, but for their brain behavior relationship within each subgroup is quite different. The relationship pattern in S1 almost resembled its own neonatal connectivity pattern, but S2 failed. Further analysis showed that for S1, there was a significant positive correlation between the connectivity and the brain behavior relationship, and a negative correlation for S2. If we decompose the whole brain connections into within between networks, this positive correlation for S1 and the negative correlation for S2 were also observed, especially 3 networks highlighted in red box, including the default mode, front broad control, and the limbic network showed the strongest positive correlation between the neonatal functional connectivity and the brain behavior relationship in S2, while the same 3 networks showed the weakest correlation in S2. So we further group the whole brain connections into 2 sets, one for these 3 networks, the other for others. You see, the positive correlation of the dominant 3 networks is still strong for S1. This could be the explanation for higher 4-year IQ in S1 because their brain behavior relationships were consistent with the perinatal functional connectivity growth directions, but S2 not, which has lower 4-year IQ. Based on the same S1 S2 membership, we further examined the similar results using neonate specific functional atlas. This validated our findings. We have several limitations in current study. First, with a relatively small sample size, we predefined 2 subgroups at the connection level. Future study can explore more subgroups with a larger sample size. Second, we use the AL template to generate the connection measures, but we validate these results using another neonate specific functional atlas. Third, a general limitation in infant resistance diaphragm study is lack of sleeping stage monitoring. The stage difference may contribute to the identified relationship level heterogeneity. Future efforts are needed to address this limitation. Finally, in conclusion, we confirm the presence of relational level heterogeneity even in a relatively homogeneous neonatal sample. The detected 2 subgroups do not differ in functional connectivity, but differ in brain behavior relationship and the 4-year IQ outcome. The higher IQ subgroup shows brain behavior relationship that are aligned with the perinatal functional connectivity growth direction, while lower IQ group not. The 3 network set of default mode network, the limbic, and the front broad control network dominates these relationship divergences. These findings greatly improved our understanding of intersubjective reliability at brain behavior relationship level and also laid the foundation for future developments of subgroup-specific models for better behavioral prediction. Here's the end of my topic. Thanks.